Sunday webinar class. This is a 6th webinar. As I have distributed you the question, you can start to write answer for this, and you have a two option. Number one, you can start this webinar and then you can write the answer, and then you can check whether your answer is correct by playing the video. The second option is, if you think you need still more practice, you can play the video and then stop, and then you can write the answer. Then you can keep on continue until we finish this question. Let's start. So this question is based on osmosis. One hand, this is more going towards unit 3. On the other hand, it is also important for unit 1. So the basic principle of osmosis is clearly addressed in this question, and you need to know again, just to revise, if we have a solution, water potential is same inside and outside of the cell, we call isotonic. If we have a solution, water potential is higher outside and water potential is lower inside, that's called hypotonic. Then the water can move in. If we have a solution which is very much concentrated solution, just like a salt or sucrose, we call it hypertonic. In that case, water potential is very low in the solution. Then the water moves from inside to outside. And for all three, the principle is osmosis, that is the movement of water molecule from high water potential to low water potential to partially permeable membrane. So this is a very basic about the osmosis. So we proceed to the question. The effect of concentration of sucrose solution on the mass of potato was investigated. The cylinders were cut from the same potato, and had the same volume. The initial mass of each cylinder was recorded. The cylinders were left in the same volume of each solution for the same length of time. The final mass of each cylinder was recorded. The diagram shows the 5 test tubes A, B, C, D, E, and that were set up in the investigation. The investigation was then repeated twice. So here tube number A, tube number B, tube number C, tube number D, tube number E. So the sucrose concentration increasing from each test tube, and the results are given here. Tube number A, 0 concentration of sucrose, that is controlled because it's water, and then 0.4, 0.6, 0.8, 0 and 1. So sucrose concentration increasing in the tubes. Mean initial mass of potato cylinders measured in gram. The final mass of potato cylinders again measured in gram. The test tube E, the range of the initial mass was 2.79 plus or minus 0.0 gram, and the range of the final mass was 2.01 plus or minus 1.6 gram. Right, what you see in the numbers, the mean value and standard deviation. So anyone still unclear about the standard deviation and calculation? I have a previous webinar, which specifically address what is the standard deviation, why it is important, how we calculate, and how we analyze this statistically standard deviation like things. I can still post this video again. If somebody wants this video webinar, I can send this one. So then again, what it says actually, if you plot a bar graph for this, 2.79 mean value is this, and then in this E tube the mass decrease. So then I can show this as like this, 2.01. So these are the two mean values. The standard deviation is how the other values fluctuate or vary with this mean value in this group. For example, when we repeatedly measure this one, we get several values and we calculate the mean value, and the highest value is 2.79 plus 0.04. The lowest value is 2.79 minus 0.04. So within this range, the values are fluctuating. That means here is the mean value, and the values are fluctuating within this mean value. Same for the other one, 2.01. The highest value is 2.01 plus 0.16. The lowest value is 2.01 minus 0.16. Calculate the largest percentage change in the mass for potato cylinders in 1 cm cube. Right, so I will show you the largest. So if we take this value and we take this value, so we can get the largest difference. So this is the largest difference we get if we can get these two values. So percentage calculation, you have to take these two values if you want to see the largest percentage change. So now I use these two values to calculate the percentage change. Again, if you are not sure or you forgot what is the standard deviation, what is its range, how the range comes, why it is important, all these things you can check from the previous webinar. Right now, we go to the calculation. So I write this first one, 2.79 plus 0.01, 2.83. 2.01 minus 0.16, 1.85. Then I have an initial value. So this one is my initial value. This one is my final value. And percentage calculation, the difference of these two values is divided by initial value multiplied by 100. So here's the difference divided by the initial value multiplied by 100. 34.6 is the answer. Okay, we proceed to the next section of the question. Explain the changes in mass of potato cylinders in test tube E. So try to explain it now. So what we see, 2.79 and 2.01 meaning mass decrease. Why mass decrease? Then it should be the water should go out by osmosis. And why we see highest change? Because 1 is the highest concentration. One is the highest concentration, so we can observe the highest percentage change, 34% mass reduced. According to the previous calculation, 34% mass reduced. Right, now 2 points. The change, the highest change I have to explain from this, and then what happened to lose the weight I have to explain using osmosis. So these two facts I placed here. Okay, that's again the same thing I will explain. That's a cell, that's a solution, and this is the highest concentration. So then water is going, because this solution is hypertonic, and the water is going out by osmosis from high water potential to low water potential. Right, so this is my model answer. The potato experienced a reduction in mass due to the loss of water through osmosis, where water molecules migrate from an area of higher concentration within the potato to an area of lower concentration in the test tube. The potato in solution E exhibited the most significant loss of mass as it was placed in solution characterized by the highest sucrose concentration, resulting in the lowest availability of free water molecules. So, next, if you want to calculate so we go back to the table. Okay, if you go back to this table, now here mass increase 2.79, 3.82 mass increase, because water is a hypertonic solution and water is coming into the cell. And 0.4, what happened? Mass decrease, slightly decrease. So then again we take this one. So then water is going out. So then in between this 0 and 0.4, there is a value. So it should be then isotonic. So this one is a hypotonic. This is a hypertonic. And in between, there should be isotonic value. That means concentration of sucrose, if we make between 0 and 0.4, it should be isotonic. So when it's isotonic, the mass is not changed. There's no net movement and mass is not changed. So if we calculate, it should be same mass as initial mass. So initial mass and final mass should be the same in isotonic solution. So if you want to calculate what is the isotonic concentration, we need to plot a graph like this. Like this. And from this graph, so if we use a hypotonic, then the mass increase because what is coming into the cell. If we use a hypertonic, mass decrease because what is going out. So then there's a zero change. That means no change means zero here. The number is zero. There's no change, that is zero. And this is the concentration belongs to isotonic. So then we can find experimentally what is the concentration of isotonic solution. We have to do this experiment and then plot a graph, and then we take this intercept as the value of isotonic concentration. For this investigation, describe how this investigation could be modified to determine concentrated sucrose solution that does not change the mass of the potato cylinders. So we have to take a concentration between 0 and 0.5 because here mass increase and here mass decrease. So we need a range now that we have to expand the range. For example, 0, 0.1, 0.2, 0.3, 0.4. So we take these values, 5 values, and we redo the experiment and then collect the data, get the mean value and plot the graph, and then intercept we take where it cut. For example, here is the graph and here is the positive value, mass increase and mass decrease. So then we have a graph like this, and the intercept here shows the isotonic concentration 0.25. Right, now I going to show you the model answer for this. Do varying sucrose concentration in smaller increments range from 0 to 0.4. So according to this, we should also use these other factors. Temperature like factor should be constant. Conduct multiple trials and compute the mean value. So redo the experiment several times and then calculate the mean value, create a graph, and then concentration intervals and determine the x-axis intercept. Next question. Beetroot cell vacuoles contain a red pigment called beta-lacin. A student investigated the effect of ethanol on the permeability of beetroot membrane. Test tubes were set up as shown in the diagram, each with a different concentration of ethanol. 20 cm cube ethanol beetroot disc. The test tubes were placed in a thermostatically controlled water bath. So when you write, you should also write thermostatically controlled water bath when we want to control temperature in this investigation as 25 Celsius degree, and time is also controlled 30 minutes. The beetroot disc were removed from the ethanol. Calculate the volume of beetroot disc with a diameter of 1 cm and height of 5 mm to 1 decimal place. So I will show you this calculation. So we use this formula, and then if you want to place to 1 decimal place, so it should be a 0.4 cm cube. I think these kind of maths is not that difficult for us now. Next one. The relative color of the ethanol after 30 minutes was measured using a colorimeter. The result of the investigation are shown in the graph. Concentration of ethanol. So here they are increasing, and the pigment leak out, and this color intensity was measured using colorimeter. Explain the result of this investigation. So, Q. Explain the result of this investigation. So 2 marks, not much. Explanation we have to give. So we can say when the concentration increase, the relative color of the ethanol solution increased. So 2 marks, we don't want to, you can see 3 areas. One is initially less concentration, slow increase, but then steep increase, and then again level off at last. So that's a graph description. I think more than that, explanation is, so explanation we can say ethanol and phospholipid, membrane phospholipid, interact, and usually phospholipid are more friendly with ethanol, so they can dissolve in ethanol and alter the membrane permeability, increase membrane permeability. So ethanol can increase membrane permeability as it can interact with phospholipid molecules. So 2 marks. My model answer is the permeability of the membrane rise with higher concentration of ethanol. Ethanol interfere with the structural integrity of the membrane. Consequently, great amount of beta-line of pigment permeates to the membrane. The result is an increase in absorbance of or decrease in light transmission as measured by the colorimeter. Devise a valid investigation to compare the effect of ethanol and propanol on the permeability of beetroot membrane. So I use combs for this one. My independent variable is ethanol and propanol. I'm going to check which one has the highest permeability. So organism is beetroot. As usual, I had to take the same species, same source, same age of beetroot. At the end, I had to tell the experiment should be repeated several times, and mean value and standard deviation should be calculated, and plot a graph to analyze the result. Measurement and method. So you can briefly say the test tubes are prepared and use equal volume of ethanol and propanol, and add these cubes, cylinders, and leave fixed period of time and take them out and then measure the absorbance. Standardizing temperature, thermostatically controlled water bath, and volumes of ethanol and propanol. So these points I placed here to get the answer. The permeability of the membrane rise with higher concentration of ethanol. This one just go down. Yeah. They use some concentration for both ethanol and propanol. It is important to identify the maintained control variable to ensure the reliability of the result. For example, these are some of the control variables, same beetroot species and age. When we cut cork borer, identical diameter, temperature, time, volume, measuring cylinders we can use, like all these things we have to standardize. Use colorimeter to assess the intensity of color. Conducting multiple trials to repeating the experiment is necessary to compute the mean and standard deviation effectively. So this is my model answer for this question. So I hope you will complete the answer, and we can learn every week something like this, which will improve your exam practice skills with the time. And again, see you next time with similar series of webinar. And again, it is very important you have to complete this task, and I will check this. Sometimes if you bring this one, your folder, to the class, I can check, and it's so it's over now. So see you again next time.